So my focus today is on the entanglement. I hope that's displaying. Are you seeing that, Linda?
It's good.
Thanks, Mike.
Yes, it's good.
On the entanglement of genomic and environmental influences on traits of organisms and especially organismal fitness. And I want to talk about that in relation to two research programs. But first, the question that really launched me into thinking about this: how much genetic variation is there that can be the basis of adaptive evolution? I encountered this question articulated by Richard Lewontin shortly before I began my graduate studies, and it has motivated me to a great extent ever since. It was a compelling question when Lewontin asked it. I would say it's still an unanswered question in our field. I also encountered this statement by my doctoral advisor, Janis Antanovics. This is not an answer to this question, but it is an encouraging statement that there are approaches that we can address it empirically. Adaptation is a dynamic process, operationally definable. So with those points to launch us, here's the outline as I plan it. I want to briefly give an overview in a bit more depth of the origin of our interest in genetic variation and early strivings to understand it. Then turn to two vignettes, research vignettes from the research program I've been involved in over the years. And then take one key finding from those vignettes to revisit the history and contemplate current genomic studies of humans. So here's our motivating question: how much genetic variation is there that can be the basis of adaptive evolution? This question, of course, stems from Darwin's articulation of the theory of adaptation by natural selection, which I've laid out here, paraphrased from The Origin of Species. And I've highlighted in italics his own indication that the heritability of variation is a crucial aspect of adaptation by natural selection, a prerequisite for evolutionary change. Now, biologists at the time very quickly recognized that this was a compelling explanation for adaptation, but they and Darwin himself recognized that it was a very serious gap in the theory, in the understanding, that there was not understanding of inheritance, that he didn't understand it, and as far as he was aware, no one else did either. There ensued, through the end of the 19th century, a period of very serious vitriolic contention about the nature of heritable variation, underlying continuous traits, and also as the basis of evolutionary adaptation. And John Beatty's talk on Tuesday afternoon in the symposium was a wonderful overview of this. I also commend to you this book by Provine as a thorough treatment of that period. I just want to highlight a few key aspects of that period. And the first is this: the work of Francis Galton, who was a cousin of Darwin, who took very seriously this challenge of understanding inheritance of traits. And he invented the method that you see illustrated here, whereby he obtained measures of traits, here height of individuals, for offspring and for the pair of their an individual's parents, and plotted those points and invented the method of linear regression to evaluate quantitatively that relationship. And this was a very important first step in biometry and biometric study of traits. Then, as I'm sure many of you are aware, in 1900, Mendel's work became recognized, demonstrating the discreteness of genetic transmission, what we refer to as particulate inheritance. And further experiments very soon after the turn of the century demonstrated that an individual's genotype does not rigidly determine its phenotype. Phenotype also depends on an individual's environment in which it develops. And a whole distinct empirical approach established that when many or even just several genetic loci influence a trait, there's a wide range of genotypic values for that trait, and that many distinct genotypes can have the same genotypic value. And these points together result in a fundamental indeterminacy between genotype and phenotype. So again, very soon thereafter, R.A. Fisher burst on the scene as a very young, brilliant mathematician, theoretician, also provoked by the challenge of understanding inheritance of continuous traits, established the field of quantitative genetics, really essentially with this one massive paper deriving the theory for quantitative traits, accounting for polygenic basis of traits, and also environmental influences on quantitative traits. And in the very same paper, establishing methodology for estimating a population's additive genetic variance for a trait. And I want to define the additive genetic variance. That is, there's a formal mathematical derivation for that. It's rather complicated, but we can just understand it as the variance in a trait that's attributable to segregating alleles at all the loci throughout the genome for a population. And this quantity is especially important because it's the primary genetic basis for resemblance between relatives and for response to selection. So with this 1918 paper, Fisher provided a genetic interpretation for Galton's findings. I should say that the title of the paper is The Correlation Between Relatives on the Supposition of Mendelian Inheritance. So building that genetic understanding into interpreting Galton's findings. And he established approaches that generalize to other relationships beyond offspring and parent. These approaches treat allelic effects throughout the genome collectively in aggregate. And they also yield estimates of individuals' breeding values. So that's a term I want to establish here. That's the genetic effect of on a trait that's transmissible from parent to offspring. Again, it has a formal mathematical derivation, but that heuristic definition will do here. However, I want to emphasize that inferring the genetic contribution to resemblance between relatives crucially requires ensuring or assuming that environments affecting individuals are independent of genetic effects. Now, experimental approaches, including those Fisher developed and many developed since, ensure this condition through replication of genotypes and randomization of those genotypes over environment, which is variable at multiple scales and in various dimensions. Now, not all populations are amenable to such experimentation, and of course we can note that humans are not, human populations are not amenable to such manipulation, and therefore human relatives tend to share aspects of their environment as well as their genes. So I'll return to this point later. But As a last point in this history, I want to acknowledge that Fisher went on soon to derive the fundamental theorem of natural selection, as he called it. Here we have the symbol W, as population geneticists have used to refer to this abstract concept of an individual's fitness. But we can operationally, using Antonovic's idea, we can operationally define this as the number of offspring an individual contributes to the population. And then the average fitness for the population has this dual interpretation. It refers to the growth rate, demography, the population growth rate for that population, and also as a reflection of the degree of adaptation of that population to its habitat. We can use quantitative genetic approaches to estimate the additive genetic variance for fitness. So again, that's the variation in fitness in the population that's attributable to allelic differences at loci genome-wide. And with such an estimate in hand, as well as the mean fitness, we can obtain a prediction of the genetically based change in mean fitness from one generation to the next with this simple ratio. So with these conceptual tools in hand, let's move on to considering the vignettes that I've chosen. In the first, we were interested in addressing these questions that I've listed here. First, what is the extent of genetic variation in fitness in the wild? Focusing on this annual plant, Nemophila menziesii, which occurs in Southern California, where I was at the time. Also, how do fitness and its genetic variability depend on competitive environment? And what is the relationship between breeding values in different competitive conditions? So here you see the hillside in the natural area in the Botanic Garden of University of California at Riverside, with the campus just beyond and the San Gabriel Mountains further on. Gert Plattenkamp was wonderfully just a wonderful collaborator on this project. He and I sampled the wild population of Nemophila growing on this hillside, and in the course of doing so, observed the range of competitive environments in which we found the plant, and set up the experiment that I'll describe as follows. So there's a genetic component to the design, and for this we used a standard quantitative genetic mating design. The plant itself is hermaphroditic, but we designated a random subset of plants to be pollen parents and others to be pollen recipients, and then randomly assigned matings, as you see shown here. This is just a small subset. There were 52 paternal individuals altogether. So each X represents a full Sib family of seeds that we produced, and each column is a paternal half Sib group. So here you see the crosses being made by hand in the greenhouse. So as I mentioned, there was a wide range of competitive environments in the field. We chose several treatments to apply. One you see here is a high density pure stand of the Nemophila alone. And there were several combinations of Nemophila with this grass that's very apparent here, quite an aggressive grass that was introduced from Eurasia well over a century before this work. But there were 5 treatments altogether, 2 pure stands, a high and low density, and then 3 involving the brome. And all of these were replicated 6 times, and each family was represented, each X was represented by 1 individual in every plot. So we planted out seeds into these plots early in January when they began to grow in the field, and followed them through their lifespans. And here are the findings we obtained. So these proxies for fitness are dry mass in the dark bars, flower number in the lighter. And here you can see that there was a strong effect of competitive environment. Here is the low density pure stand and the high density pure stand of Nemophila, and the others in combination with the bromes. I'm sure it's no surprise that we could detect a competitive effect on the mean for these different proxies for fitness. But I do want to emphasize that these are genetic replicates expressing these differences. But on to the genetic variation. We did detect significant additive genetic variation in certain of the treatments. Here I'm going directly to the presentation of the breeding values for fitness. Each plot presents the breeding value for each sire for a pair of treatments. So in each case, in each plot, the high density pure stand is on the x-axis, and one of the other treatments is on the y-axis. Here, the low density pure stand in this case, and the other mixtures on the y-axis in these other cases. So I'll particularly call your attention to this case of a very strong negative relationship between the breeding values expressed in the one and the other competitive environment. So an instance of a strong genetically based trade-off between fitness, or our proxy for fitness here is the dry mass of plants in the two environments. So just to underscore the point, families that had very high fitness in the high density pure stand had extremely low fitness in this particular mixture, and vice versa. This was another instance of marginally significant negative correlation between the breeding values, and in the other two cases, essentially no relationship. But in all of these comparisons, pairwise comparisons of the treatments, we see strong evidence of the dependence of genetic effects on fitness on the competitive environment. So strong contingency of genetic effects on fitness, as evidenced in this genotype environment interaction that we detected. And I do want to note that Frank Shaw played an important role in this analysis. So now I want to fast forward 30 years and move a couple of thousand miles east and north to where I am at present. In this example, we were focusing on implementing Fisher's fundamental theorem of natural selection to predict the rate of adaptation for populations of this species, Chemicrista fasciculata, another annual species. This is a member of the Fabaceae, the legume family. It's a species that has a very wide distribution in eastern North America. We chose 3 populations to study, 2 in Minnesota and 1 in Iowa, with the help of an involvement of Vince Eckard at Grinnell College. And so we sampled from each of these populations a representative collection of individuals to grow in the greenhouse, and again do crosses in just the very same crossing design that I showed you before, the very standard nested mating design of quantitative genetics. And here's Rachel Payne carrying out those crosses. And she worked very hard herself, but with a team produced very many seeds, which was necessary because we wanted to carry this experiment out for 3 full years, planting the very same families back into their home sites in each of 3 years. So I will emphasize that these are all within population crosses, but we're studying these 3 distinct populations. We planted the seeds out in the fall when they would normally disperse. And here you see Rachel again working on the planting, along with Seema Sheth, who had joined us as just a terrific postdoc on this project. Here's Seema. Here in that first fall, we are in Iowa with Vince Eckert in the background and a team of Grinnell College students who came out to the Conard Experimental Station to help us plant. And then when Seema had to leave for other opportunities, Seema is now an assistant professor at North Carolina State University. Mason Kolbaba joined us for this project as a terrific collaborator. And Mason is now an assistant professor at Our Lady of the Lake University in San Antonio. So as I mentioned, we planted in the fall. We followed these individuals through their lifespans, monitoring them for each of these significant life stages, fitness components. We are now in the very fortunate position to be able to jointly analyze all these components of fitness, thanks to the work of Charles Geyer, who has been a really terrific collaborator in deriving the statistical theory and then implementing it into software for what we call Aster modeling of fitness. And this is the graphical model for fitness, in this case, a relatively simple model, linear dependence of each life stage on the previous one. And we've shown here the sampling distributions that we assume for each transition from one stage to the next. So on to the results. For each population in each year, we detected highly significant additive genetic variants for fitness. Here, I'm displaying for each population the estimated distribution of breeding values in each year. So 3 distributions representing the different years. So we have gone on to make the predictions of the genetically based change in mean fitness using Fisher's fundamental theorem, using this data set. I refer you to this paper to learn more about that. I'm very excited about that aspect of the project. But for this talk, I want to focus on this finding that we found highly significant interactions between genotype and year for every pair of years for each population. So let's look at that. These plots are just very similar, just analogous to the ones I showed you before. In this case, each row refers to a population, and each column refers to a pair of years for which the breeding values are being compared from one year to another for that population. Now, we don't see any negative associations between breeding values from one year to another in this case. But as I said, there is highly significant genotype by environment interaction. And there are quite a few instances that I think you can see here of cases of a family that attained quite high fitness in one year and yet very low fitness in another year. So these first 2 columns are the adjacent years, and then this last column is 2015 versus 2017. So in sum, by treating the adaptive process as operationally definable, we respond to Lewontin's question, how much genetic variation is there that can be the basis of adaptive evolution? that there's substantial additive genetic variance in fitness in populations, even though these are populations growing in their home sites. And this implies that there's capacity, substantial capacity for ongoing evolutionary adaptation. And I want to emphasize that Fisher himself conjectured in his book immediately after the derivation of the fundamental theorem that there would be very little additive genetic variance for fitness in wild populations. And this is a view that is really quite widespread among evolutionary biologists. But here we see quite strong evidence that it is, there is additive genetic variance for fitness. But going onward, we've also found profound contingency of genetic effects on environmental conditions. And we can break that down into two aspects: the overall effects of environment on the average fitness of a population, of genetic replicates of populations. They differed among biotic regimes, as you saw in the first example, and also among years in the second example. And we did no manipulations of those environments in the successive years. But besides the overall effects, there is just this superabundant evidence of interaction between genotype and environment, the contingency of genetic effects on fitness, such that differences in fitness among genetic families relative to each other vary from one year to the next. And so evolutionary adaptation in the wild under the vagaries of environmental conditions can indeed be highly dynamic, as Entanglement suggested in that quote I had. So before I move on, I want to acknowledge that the evidence of dramatic environmental dependence of genetic effects, it's super abundant. There are many examples. I have favorite examples from the agricultural literature, lab studies, and also from many colleagues who work in the field. I have no ownership of having shown genotype by environment interaction. I would say it is relatively rare that a study has completed inferences about fitness taking into account all the components of fitness through the life cycle. But this is a well-established property, and it doesn't refer to plants alone, as I'm sure many or all of you are aware. I also want to emphasize the aspects of experimental design that were required to achieve, make these findings. First, genetic replication at multiple scales, in this case, spatial and temporal scales of environmental variation. And also, very importantly, randomization of individuals representing each family over the fine scale environmental variation. So together, these aspects of experimental design prevent confounding of genotype and environmental effects, such that those main effects of environment are distributed among all of the individuals. And these properties also enable direct study of the interaction between genotype and environment. So with these points in mind, let's return to Francis Galton. So as I said, he established the quantitative study of inheritance. And then we have this interpretation from Fisher that given the degree of relationship between pairs of individuals, and hence the proportion of genome they share, the extent to which genetic variation contributes to trait variation is reflected in the degree of resemblance. Now, inverting this logic leads to our approaches to estimating the genetic variance for a trait, under this crucial assumption that nothing besides shared genes contributes to their resemblance. Now, Galton studied a wide range of traits, including measures of genius, as he referred to it, and various abilities and social standing, eminence. But let's pause here a moment. If I can advance my slides. To think about the meaning of these words, heritable and inherited. So in our research community, they just routinely refer to the process of genetic transmission whereby offspring receive from a parent alleles embedded within a full genomic complement, and consequently offspring trait values similar to parent trait values. But of course, these are common language words, and I learned that the first use in English is traced to the 14th century. And here's a definition: received from an ancestor as a right or title, descendable by law at the ancestor's death. And of course, we still use these words in outside the context of genetics. So any property offspring receive from their deceased parents. So Galton, intent on addressing the key gap in Darwin's theory of adaptation by natural selection, attributed the resemblances between offspring and parents that he found entirely to genetic transmission, ignoring other modes of inheritance. And he inferred genetic determinism, without using that phrase, of many human traits, including intelligence, ability, eminence. And he coined the term eugenics and advocated eugenic programs, which is to say breeding by and between people of high attainment. And many endorsed Galton's promotion of eugenic policy then and in decades following, with profoundly tragic consequences. But there were important exceptions, and one was Alfred Russel Wallace, and I really love this phrase I found quoted of his: eugenics is simply the meddlesome interference of an arrogant scientific priestcraft. And I think this is a wonderful early example of a point that Butch Brodie made yesterday in his ASN presidential address of the value to our whole community when access broadens. So Alfred Russel Wallace was himself not particularly highborn, certainly compared to Galton and Charles Darwin himself. But by this time, when he was quoted, he had achieved quite some standing as a biologist, of course, having independently inferred the theory of adaptation by natural selection and carried out much other important work. So I've benefited a lot from conversations in contemplating this period, from conversations with historians and philosophers of biology here at the University of Minnesota, along with my evolution colleagues. Here I want to refer to a paper from a historian of biology, Garland Allen, who about 10 years ago was grappling with this period to glean lessons for our present time. And I'll quote pretty heavily from this paper. He noted that prior to 1925, there was little open published criticism, and what there was remained largely in academic journals. But after that, considerably more criticism was published by well-known geneticists, and you see a partial list here. And this was important because it signaled that eugenics did not have the unqualified support of the entire genetics community. And he goes on later in the paper to say he finds two important conclusions to emerge. And one is that knowledgeable geneticists should examine claims about the inheritance of traits when they are publicized today. And the conclusion of this quote, since claims about the genetic basis of such characteristics can have serious medical consequences for individuals, and of course, as we know, very serious societal consequences. It is important that scientific accuracy of the various claims should be clearly delineated and exposed. And his second point is that geneticists should also make their critique public and not restrict their publications only to technical journals. Okay. Now, I want to acknowledge that many others, evolutionary biologists, historians, and philosophers of science, have emphasized entanglement of genetic and environmental influences on traits and the speciousness of genetic determinism. And here are just several books I find really worthwhile in this regard. I also want to note this paper by Josephine Pemberton, among many I could cite, but Josephine has devoted a wonderful career to studying mammals in the wild. And in this particular paper, she really grapples with the challenges of making genetic inferences about populations in the wild when it is not really possible to do the experimental manipulations. Despite all this, efforts to find genetic determinants of human traits continue apace, with only slight mention of environmental influence. And these days, the approach that's used is genome-wide association studies and polygenic risk scores. And this approach is very closely aligned to the approach of Galton, but just to give a little overview here, the approach relates trait similarity between individuals to sharing of nucleotide sequences at many positions throughout a genome for individuals that, in this case, are not closely related. And the approach also involves accounting for patterns of sequence similarity deemed to reflect population history or structure that could otherwise be misleading about the genomic basis of traits. And I quote here from a review of the first 10 years of genomic studies of human populations. GWASs have shown that the same genetic variants can be significantly associated with multiple diseases and traits when phenotypes are measured on unrelated individuals. And here I want to emphasize the parenthetic phrase at the end of this sentence, so that no environmental associations are driving the results. So I want to emphasize that this is an assumption of the methodology, that because unrelated individuals are being compared with respect to their genomic sharing and trait sharing, it would be impossible that there could be environmental conditions that could drive the results. And here's an example of a paper in this vein where the authors, this is the final sentence of their abstract, say that in modern era Great Britain, genetic effects, according to their study, contribute toward observed socioeconomic inequalities. That's from the abstract. In the discussion, they do note that the current data cannot differentiate between direct genetic causality and dynastic effects. So here they actually acknowledge that both genetic transmission, but also this other more common language mode of inheritance could be contributing to their findings here. Not what we see in the abstract. And claims of papers like this are brought to public attention. And here are just a couple of examples, one from the newspaper of record, as it's often called, the New York Times, and one from the Minneapolis Star-Tribune. So as we, in the present time, reflect on the painful history of eugenics in evolution and genetics, And I'll acknowledge that it's painful to me, but I know from conversations with many of you that I'm not the only one who feels that way. So we have this painful history, and we have the persisting societal legacies. We must acknowledge that we can't change the history, but the question is, can we learn from it? And how can we as evolutionary biologists act? And Alan has a recommendation, and that is that those with the expertise to expose the fallacies in oversimplified genetic explanations should do so to ensure that the misuse of our science does not occur again. I'm happy to say that in the 10 years since Alan's paper was published, there are some examples, and here is one from Graham Coop's group, where in the abstract we read, "We find evidence that previous analyses were confounded by population stratification. Therefore, the conclusion of strong polygenic adaptation," that's for height through latitudinally through Europe, "now lacks support. And these discrepancies highlight that methods for correcting for population stratification in GWAS may not always be sufficient for polygenic trait analyses, and that claims of differences in polygenic scores between populations should be made with caution." I can also say that to some extent, these challenges are being brought to out in more accessible forms. This is a blog post by Graham himself that unpacks the work in a very nice way. And also this brief paper that was published together with the technical paper I showed in the previous slide. And in this, we read from Barton and colleagues, "The issue is that there is no perfect way to control for population structure and environmental heterogeneity. But much more needs to be done." So I leave you with these key points. First, there's substantial capacity for ongoing adaptation in the wild plant populations we have studied, and I hope other studies of this kind will be done in the future. We've also found that the adaptive process is highly dynamic due to environmental variability, along with interaction between genotype and environment. And again, to underscore this point that validly discerning genetic effects amid ubiquitous environmental variation requires experimental replication and randomization of genotypes. And finally, inferences from non-experimental GWAS, and especially those of human populations, are severely problematic. First, they are unsound for the reasons that were articulated in the previous slide in the Barton paper. And in that way, they undermine the credibility of science. But even alongside that, even more important, they pose the clear risk of individual and societal harm. So I just want to urge us all as a community of evolutionary biologists to critique the claims, because we who understand genotype by environment interaction, confounding of genetic with environmental effects, are best equipped to articulate those challenges. And with that, I'll conclude. I've noted the people who had direct involvement as I went through. I've been so fortunate to work with so many wonderful, effective, capable, and clever people over the years. And these are just to signify that. I also want to thank those who listened to my talk and gave me wonderful, constructive comments, colleagues here, as well as kids of mine, and funding from the National Science Foundation. And with that, I will be very happy to answer questions and comments.